A very good morning to everyone. Thank you for taking time and joining our webinar 11, for which we have Dr. Uday Saxena with us. So before we start, I would just like to ask Dr. Surat, the Senior Research Associate at the Center for Predictive Human Model Systems, to just give a brief about the activities which we carry out at the center, followed by which we'll have a talk by Dr. Uday. And if there are any questions which are there during the presentation, you could type in the chat box and we would take it after the presentation. Having said that, so I would now request Dr. Surat to start a presentation.
Thank you, Sham. So I'll just take a few minutes of your time just to briefly talk about the center and why we are having these presentations, these webinars. Sorry. Can you see my screen?
Yeah.
Yes, Doctor. Yeah.
So as Sham mentioned, I'm Surat. I'm a Senior Research Associate at the Center for Predictive Human Model Systems at Atal Incubation Centre, and this Science and Policy Centre that was established in collaboration with the Atal Incubation Centre and CCMB and Humane Society International India to enable more research that is based on human cells and is more relevant to human biology. So just to give a brief primer about what do we mean by human relevant model systems. So while animal models have given us extremely useful insights for past several decades, the past 1 or 2 decades, we've also, researchers have also developed several model systems that are based on human cells and human biology, such as organ-on-chip, organoids, 3D engineered tissues, ex vivo cultures, etc., which can generate data which is more relevant to human biology and thus can capture, aim to capture the subtleties of human biology. As a center, we have been, our aim has been to enable more of this research in India, and towards this, we have been focusing mainly in 4 directions. So one has been increasing awareness for these new and emerging methodologies in India. And this has been via popular science articles, reviews. This webinar is another such example to increase awareness. Another such initiative is we also conduct several educational and training workshops, and we'll be announcing an India EMBO lecture course soon in this area. We've also involved in several policy initiatives in the sense that we've written several white papers to understand what are the challenges and to conduct this research in India. And we also recently conducted a multi-stakeholder roundtable meeting to provide recommendations to the government bodies in terms of how these challenges can be addressed. And we're also involved in several global collaborations, again, to further address both technological and regulatory challenges currently present in this field. And in terms of funding, our center, in collaboration with Humane Society International, also recently provided a grant to 2 Indian scientists to develop an adverse outcome pathway in the field of cancer. And just to kind of end on the note that what we perceive as a roadmap to enable human relevant research in India. So the few ways via which we could do that is, one is by establishing centers of excellence to further develop and validate these methods. We would like to enable connections between technology developers in academia and industry and end users. We would like to engage with regulatory bodies from the initial development of the technology just to ease the process of regulatory approval. We would also like to further enable cross-border collaboration, both for technology transfer and for international funding. And another issue is we'd also like to further enable supply chains for various aspects such as reagents, tools, etc., for indigenous development of this technology in India. So with this, I would like to end, but just a brief heads up that we are also planning to launch a newsletter this month. And this would be a one-stop place for all the human relevant research, but also informational videos, opinion articles, publications, and also possible grants and job opportunities in this space. And we'll be posting the link for registration for this newsletter in the chat box. And please do register for this newsletter if you're interested in this space. And second is towards the end of the talk, after Dr. Uday gives his talk, we're also conducting a small poll on what is the awareness for good in vitro method practices in India. So while as we are developing more complex in vitro methodologies, there is also a parallel need to develop standards and guidelines in terms of how do we use these methods, how do we handle, store, and also report all these stem cell based new model systems that we are developing. And OECD and several international bodies have also come up with several guidance documents in terms of what are the proper good in vitro method practices. So good cell culture practices, GCPP 1.0 was mostly based on in vitro cell culture systems, and now that concept is being expanded to incorporate microphysiological systems. So it should be GCPP 2.0, and we are conducting this small poll just to understand what is the awareness for these cell culture method practices in India. So why are we concerned with having followings, international guidelines or protocols? So one would be to having a specific set of guidelines which is followed both internationally and in India, would help to increase more widespread adoption of these methods both in industry and in academia, to increase reproducibility. If you are both developing and following a standardized set of protocols, would help to increase the reproducibility of these new technologies, and also to increase trust amongst scientists, industry, and policy making bodies for these new methodologies. So please do stick around till the end for this small poll. It's not going to take much time. So with this, I would like to end and hand over to Shyam for introducing the speaker for today. Thank you.
Yeah, thanks, Dr. Surat, for the presentation. So moving on, so now I would like to invite Dr. Uday for the 11th webinar in the series, who will be speaking on in vitro humanized disease models, focus on 3D bioprinted lung models. So that is the topic. And just briefly to introduce Dr. Uday, Dr. Uday is a mentor in residence, also a professor of translation research at Dr. Reddy's Institute of Life Sciences. Apart from it, he's also the co-founder and the chief ideator of Reagene Biosciences, biotech startup based out of Bangalore and Hyderabad. And he has also held executive and leadership positions at Parker Davis and Dr. Reddy Labs and Caris Therapeutics. So he has vast experience in dealing with drug candidates and bringing them from the idea stage to the clinic. He has also received his PhD in biochemistry from the Memorial University, Canada, and postdoctoral training at Columbia University. And his research largely is based in metabolic diseases and cancer. So he is the entrepreneur of our science, where we can see that he has taken the research which happens in lab successfully into the market. So we're glad to have Dr. Uday with us today. And over to you, Dr. Uday, with the presentation. Just another thing, if the audience have any questions to Dr. Uday between the presentation, so you could type it in the chat box, and then we will take it up after the presentation. And also, please just stick on till the end for the poll which we have. Having said that, Dr. Uday, over to you.
Yep. Thank you very much. First of all, Shyam, for that very generous introduction. You know, it's always nice to hear other people say good things about you. You never get tired of that. So I want to thank Surat for the invitation, although she waited for a year to bring me on here. So, but finally, I think she saw the light and she's invited me. Very happy to be here. Also would like to acknowledge meeting some good friends, Dr. Radha, Dr. Suresh, and Brinda. These are all people who, you know, I have interacted with and have very good memories. So thank you guys for taking out the time to be here. I will now try to project my slides, which sometimes is a challenge. So just give me 1 second. Can you see my slides?
Not yet.
Okay. Oh, did you do the share? Sorry, I have to go back and see if you did if you allowed me to share the slides or not. Did you allow me?
Shyam, is the option enabled for him?
Yes, Dr. Uday. Yes,
okay,
Dr. Uday.
All right, now let me try it then.
Yeah.
Now you can see the, okay, now then I have to. Yeah, again, what I'm going to do, thank you, Shyam. is basically tell you about bioengineering of alternate models with a specific focus on 3D human vascular lung model. This is something we developed over the last 1 year, and very excited about it. And I'll actually share some data on how we have used this model to identify some new drug candidates for COVID-19. So I don't know why. Okay, let's give me. Yeah. So as the presentation points that I'm going to hit upon is, first of all, why is there need for alternate models? And secondly, then I'll tell you what is the global status of alternate models. A lot of work is, as Surat said, a lot of work is being done in alternate models in this field. And then I'll tell you about how new 3D models are being developed. Then I'll move on to tell you about our own human vascular lung model and some of the data that we have generated, and finally end with conclusion. So that's okay. So first of all, let me, I'm trying to minimize. Sorry. Yeah, so let's look at the need for alternate models. Why do we even need alternate models? And when I say alternate, it's alternate to animals, obviously. So if you just look at the graph here, or what I have, the graphic that I've shown, you will see that when you have to take a drug forward from an idea into the clinical trials, it has to go through an early phase called discovery, then preclinical. And then you're allowed, once you have to then file something called an IND with the FDA, and then you're allowed to do clinical trials that are phase 1, phase 2, phase 3. The problem here is bulk of the work, 99% of the work that you submit to the US FDA to allow you to enter into clinical trials is all done in animal models. So that becomes a big black box, because here you're hoping that you'll be able to mimic some of the human diseases, the safety, the toxicity in an animal model. And as I will show you in the next slide, that really is not been turning out very well. I mean, the attrition rate is very poor. So that suggests that the animal models are not being being very predictive. And the only reason we are using animal models today is because we don't have anything else. Literally, we don't have anything else. So the FDA, it has to have some understanding of what the toxicity could be. So they'll let you do it in rats and dogs, is really the two species. But there is clearly a need for introducing new, more human-like models to be a better predictor. So this is a case in point. If you look at the attrition, when you start a drug discovery program, you start off looking at one target and maybe 5,000 to 10,000 compounds that you screen against them. And during that screening process, about, as you can see, 90% of them drop out. You're left with about 250 drug candidates that are there. And once they move into the clinic, the number gets even smaller. So it gets to about 5 in phase 1. And by the time you finish phase 3, it's just 1. So humongously huge attrition is what you get to see here. And a large part of this dropout rate or attrition is because you're not using the right humanized models. It's largely because using the rat and the dog to be able to predict what's going to happen in humans, and that results in this attrition. So there's a very strong reason now, and the industry has realized it in the past 7, 8 years, I would say, that there is a need for alternate models. And the FDA also, I think Surat talked about maybe, I don't know, maybe not, she didn't. But already, if you have to develop some cosmetics, for example, the US FDA will not allow you to use animal models at all. And that's going to become the norm. Eventually, a lot of the regulatory agencies are going to move away from using animal models and use better humanized models. The only reason we are not using humanized models today, or human models, like models, is because we don't have them as good as they really need to be. All right, so there is a clear need, and the future lies in creating human-like models. There is absolutely no question. all the regulatory agencies are heading in that direction. So people have understood this, and it's been about 10, 15 years that people have started to think about journey of alternate models. And if you look at, just focus on the second column, right? So about 25 or 30 years ago, we were very happy with being able to culture cells in static flasks, right? That was the initiation of trying to mimic human biology in a dish. And we are very happy with that. Then we moved on to what's called a co-culture. So you just don't have one cell, so you have 2 or 3 different cells being grown together. And so the idea is to try and recreate an organ. Then came the dynamic cultures. And then more recently, I would say more like in the last 5 years or so, organ on a chip, which is like a very micro version of what you want to do. If you want to go a blood vessel, and I'll give you an example, you could do that, and you're dealing with like 1 or 2 microliter types of volumes when you do with an organ on a chip. And then comes 3D bioprinting. I think the last 3-4 years, and I'll just show you at the bottom, there's a graph there. You can see how there's like a huge explosion of 3D bioprinting and its use in research, both for drug as well as basic research. And what I wanted to share with you, and this is because of the data that is being generated. If the left-hand bottom, you'll see there's like a rat and there's a blood vessel being shown there. That's a morphology. There'll be endothelial, smooth muscle cells, and a very nice lumen. You cannot get to that using an organ on a chip. The organ on a chip on the second at the bottom, you'll see, doesn't quite have that morphology there. But if you look at the very bottom, that's a 3D bioprinted. The morphology, and has been shown, the physiology of these 3D bioprinted is very similar to what happens to an actual blood vessel. So I think there's a lot of agreement now that 3D bioprinting potentially is the way to move forward in creating human types of models. And that's being reflected in, as I shared in the graph, that there's a huge explosion in the use. So what are the 3D printing approaches? So 3D bioprinting, I have a video, but I'll only show it at the end if we have time. But other than that, I mean, Shyam has this presentation and he can share it if you want. Nothing here is proprietary. So if you look at the very left, 3D bioprinting involves basically 3 things: the cells that you want to print. You have to have a material on which to print the cells. These are called either biomaterials or bio ink, I mean, those are different names for it. Then you have to have an actual 3D bioprinter. I'm not showing you a picture of the full printer here. All I'm showing you is the types of delivery systems. So the cells and the matrix is mixed in and put into these syringes, which is attached to a 3D bioprinter. The 3D bioprinter is actually programmed to dispense cells at a certain rate, at a certain pressure, in a certain format. So it's very high precision. One of the reasons why I believe 3D bioprinting has overtaken other types of in vitro technologies in trying to mimic human, I think, is the precision, the reproducibility, and the architecture that you can create with 3D bioprinting that you cannot do it with other technologies. So there are 3 types of bioprinting. One is called inkjet. Again, this is how you dispense the cells. We normally use inkjet, but there's also an extrusion base, which is shown in the middle. And there's also more complicated, like the light-assisted, laser-assisted. So these 3 are the basic fundamentals of a 3D bioprinting. And once you have your 3D bioprinted tissues, you have to do some validation. And on the right-hand side, you will see you have to make sure that the morphology, if you're trying to do a blood vessel, right, there's a certain morphology. So you have to demonstrate that the morphology you're creating is actually what you see with your 3D bioprinted also. So a lot of morphology and functionality has to be done. And typical users of this shown at the bottom is trying to look at drug-induced toxicity. Are we able to recapitulate the toxicity that's being seen in humans using this model or not? So a lot of validation. We spent, I started 3D bioprinting at Reagene in November of 2018. And I'll tell you, it's only that last 1 year that we have gotten the whole thing together and are able to now successfully generate data and, obviously get patents and publish. So it's not something you can just get up and go and just say, okay, I see materials and methods. They say, do this, do this. It's not as simple as that. Every user's experience is different. It depends on what cells you're using. It depends on what biomaterial or bioink you're using. And finally, it depends on which organ you're working. Some are easier to do than some are more complicated. Okay, so the typical uses of 3D bioprinting fall into like 3 buckets. At the very left, as I've shown, is bioengineering. So these are usually typically bioengineers who want to create the Taj Mahal of an organ. So they will tweak everything from cell morphology to the biomatrix, the viscosity. I mean, these guys play with that with the ultimate aim that they will be able to get to the Taj Mahal. So a second set of people are actually more translational in their approach. They want to recreate human organs, either for research, such as liver. Dr. Radha and I were just talking about her trying to recreate a liver, right? So that's a fantastic use of these technologies. And the idea there is either you can use it for research. For example, liver is very important in drug metabolism. You can use it for that. Or ultimate, the holy grail here is that you should be able to use these homegrown 3D bioprinted organs for transplant. So people now have to wait for liver transplant for a few years. So potentially, if you could do this, at least in the interim, while the patient is waiting for a donor, they could try and use this. So that's another thing that's happening, which is very exciting. The last thing is people like me or people like us. We are trying to build disease models. Can we recap the disease that you see in humans using a 3D bioprinted or not? So from the industry side, that's of immense interest because, and I'll tell you how it came very handy for us while working in COVID. But so the green bucket is where I am working on, and I'll share some information and data. There is a 3D bioprinting video how-to that I'm not going to share now. If time permits, it takes about 5 minutes, so I'll just leave it for now. We'll do it later if there's time. So why did we get into developing a human vascular, 3D bioprinted human vascular lung? We got interested prior to this. We were mainly trying to recreate a drug metabolism system using liver cells, intestinal cells, and target organs and all that. So that's what we are working. But when COVID came, as you know, about 8 out of 10 scientists, I think, got interested and started to work. So we were no different. So we started this work about a year ago, I think. And our basic goal was to recreate a human vascular lung. And I'll tell you what that means. And the reason for that was that there were no good animal models. A year and a half back almost, there were not any good models. Even today in India, it's not easy or inexpensive to go and get an animal model. There are two basic, the hamster model is there, which is really a 5-day model and does not recapitulate some of the important things that happen in real life. And there's the human ACE2 lung overexpression model. So those are the two, very hard to get and not fully validated. So that's why we said, if we want to find new drugs, and our approach was not to look for brand new drugs, but the FDA has approved a lot of drugs in the past. And so can we find one of those that we could actually test in our vascular model? And if it looks good, then you can move it forward for COVID, because if it's already been approved by US FDA, it means it's safe, right? And secondly, it also means that it'll be available to people. So that's the reason we went for repurposing of drugs rather than trying to find new drugs. And our first step in that journey was to create a human vascular lung model that creates the elements of COVID. Okay, as you can see here, this you may have seen. I'm just trying to give you the the shock and awe version of what physicians see with the lungs. Left-hand side is somebody, you can see the lungs are pretty clear. There's hardly any there. But the next two are medium and high degree of COVID. You see this typical image, the fogginess is also called as shattered glass. And that's because the lungs are now full of fluid, full of virus, and full of inflammatory cells such as monocytes and neutrophils. That's what gives it this. So it's pretty dramatic. And you can imagine when the lung is in that situation, it is not able to function well. And that's when the breathlessness comes in. So, but behind the images that you see here, there are certain very critical molecular events that happen. So what exactly are those? All right, so what I'm trying to show here is the basic involvement in COVID is the lung, as we all know, that's the target organ, although other organs are equally hit, but it's the lung that gets impacted the most. The virus enters even through the nose or the oral cavity and then finds its way into the lungs, right? So what I'm showing here are two cell types. One is the lung, the alveolus that you see there is nothing but a sac that's made up of lung epithelial cells, shown there in blue. The other thing I'm showing you here, that pipe you see, that pipe is nothing but a blood vessel wall or an endothelial cell. So the two critical cells that are important in COVID is one is the epithelial cells of the lung, because they form the barrier to allow or not allow things. And the other in close proximity are the capillary, blood vessel capillaries. You probably know this, but the lung is the most heavily vascularized organ, which means it has the highest number of blood vessels of any other organ, right? So it obviously, because oxygen handoff and carbon monoxide, all that happens because of the blood vessels and the alveolus. So these are the two cell types that are important. And so there are three critical things that happen. As soon as the virus enters the lung cells, it has to cross through the endothelium and then get into using the RBD domain on the spike protein and the ACE2 receptor on these cells. It gets through in. Once the virus gets in, the lung sends out a message saying, we have a foreigner here, we need to get rid of it. And that's when inflammatory cells that are shown here, you can see in purple, they start to arrive. And their modus operandi is two. One is to secrete cytokines in the hope that these pro-inflammatory cytokines will create an inflammatory defense mechanism to kill the virus. And the other thing that they can also do is they can actually phagocytose the virus also. But majority of what you see is the release of inflammation. So three things have happened. The virus in the blood has gotten through the endothelium, which means it has probably done some bad damage to endothelium also, then gotten into the lung using the RBD domain and the ACE2 receptor, that's the viral entry. Then because of this, inflammatory cells have come in. And then finally, you see a huge explosion of cytokines being released, which is also known as cytokine release. So these are the three critical events. There's one more that I'll tell you about that needs to be captured in any human-like model that you want to study. All right, so that was kind of what we're going for. So the first thing is, let me tell you a little bit about the inflammation that you see in the lung. On the left-hand side, you will see there's like a blue swimming pool kind of thing. So that's nothing but a lot of edema and fluid that has gathered in the lung. And in the middle, you will see macrophages. These are monocytes that have come in from the blood to try and help the lung to fight off the virus. And then there's also the neutrophils. So the lung and inflammation are the first signs of COVID trouble. As soon as the virus enters in, the lung kind of gets into SOS mode and starts to attract these defensive cells and try and get rid of the virus. So that's one thing you must actually be able to recap. The other thing is, again, I have three compartments here. On the very left is the green one, which is, either a normal situation in which normally you shouldn't find any cytokines in the lung at all. In the middle is like a medium where the COVID has come in and the lung is trying to fight. But if the number of viral particles is huge, then the persistence of the macrophages and all, which actually secrete cytokines, becomes like a vicious cycle. That's the very last. There is uncontrolled over secretion of cytokines in the effort that the lung is making to try and get rid of this virus. So if the virus, as it replicates more and more, the cytokine storm gets worse and worse, right? So it's like a vicious cycle. The cytokines are good in the early on, but once we have too much of them, they pretty much light up the lung. I mean, they initiate so many different molecular pathways, and it basically goes out of control. So just like inflammation, cytokine storm is also not a good thing. And so that's the second thing we tried to capture in our vascular lung model in our hope to recap a human situation. The third and very, very important thing that people think might actually be the big reason why people die, of course, the lung dysfunction and breathlessness and lack of oxygen is one. But the other thing people are seeing is what's called thrombosis. So again, I have on the left-hand side a lung that you can see at the top, the blue lung. And if you see a cross-section of the lung, there's not, especially in the middle, you see, this is a blood vessel you're looking at that supplies, oxygen and nutrients and everything to the lung. You'll see that it's pretty squeaky clean. It's like a pipe where the water can easily flow through. But if you look at the right-hand side, it's the same thing, but this time it's with a COVID patient. And you can see there's a lot of thrombus formation. Thrombus is nothing but once the endothelium is activated, and the cytokines we talked about actually are very good activators, they create a very thrombogenic surface, which means platelets start to bind and aggregate. And as a result of this thrombus formation, it's really a clog that does not allow blood to go through. So you can imagine if this happens in critical organs, such as like the heart, you will obviously have cardiac arrest, and it can happen in kidneys and all that. So it is now being recognized that thrombosis is actually a very severe consequence of COVID infection. Previously, it was just thought it's inflammation, cytokine storm, but as physicians understand the pathology better, it's very clear that thrombosis. So this is another thing we wanted to recap in our 3D lung model. So how did we create our 3D vascular model? So as I told you, the two important players are the lung endothelial cells, and the second is, the lung epithelial cells, and then the blood vessel endothelial cells. So we decided to recreate, and that's why we call it a vascular lung, because you now have blood vessels too. So we created a lung epithelium to recreate the alveoli, and then we also, underlying that, we also had the endothelial cells to recreate the blood vessels. So you can see that there. And at the bottom, you can see pictures. At the top is the epithelium, which is a lung epithelium, and then there is a basement membrane, which is basically in 3D bioprinting. It's the matrix that we use. And then there is the endothelial cell. So all these three were bioprinted using a bioprinter that we have. And we actually, you can bioprint in 6 wells or 12 wells or 24 wells, but we have actually designed our own robotic 3D bioprinter. We can do all experiments in 96 wells now. Beautiful bioprinting in 96 wells is what we can do, which allows us to do many more things rather than have to rely on, a 6 well or a 24 well or a 12 well. So that's the model we created. This is more how exactly we did it. We first printed using the 3D bioprinter. We printed a layer of biomaterial, such as a matrigel. Matrigel is nothing but something that's, it's actually largely collagen and proteoglycans. This is basically a endothelial cell matrix, right, that occurs in real life. So for your cells to be happy, it's important to have that. So we bioprinted that using our bioprinter, let it sit for a while, and then we print the human cells. In this case, we use lung epithelial cells, okay? Then you let it sit for a while. Then you then have another layer of the biomaterial or bioink, and then you let it sit for a while, and then finally you add the lung cells at the very top. And what you get at the extreme right, you get this a snapshot of a 96 well, where you have the biomaterial, you have the blood vessel cells, that's why it's called vascular lung, then you have the lung cells. So this is the system we created. And to the best of my knowledge, there's not many people, if any, who's doing this in a 96 well format. I mean, people have been doing it, but they're mainly bioengineers who are trying to create the Taj Mahal. Our interest more is in, can we create a reproducible system? See, one of the things when you work with drugs is, today, if you find that the drugs were in a certain way, say the IC50 of the drug is 5 micromolar, right? When you do it again the next week, it shouldn't go from 5 to 50. That is not a tool that we can use in drug discovery, because we make very important decisions on which drug to move forward with based on such data. So speed, throughput, as well as the reproducibility of the data. And this is why I like the 3D biosprint system. And I've been working almost now 2.5 years. Reproducibility of the system is just perfect. And I think it comes from the fact that you are having an architecture that's been robotic printing of an architecture. So you get a precise layer by layer. And I think that's really what the reason, and so do other people. So now we created the lung model, the vascular lung model, which has the blood vessels and the lung cell. What do we do next? Okay, in this model, as I said, we first had to validate a few very important things that we saw in that is being seen in human biology. First, we should be able to score for the entry of virus into the lungs through the binding of RBD on the spike protein on the virus to the ACE2 receptor in lung. So we were able to do that. I mean, we created a system to be able to do that. And basically, you can use antibodies against the RBD or the ACE2 to try and block, and that's one way you can show that, okay? Then we created a system to look at inflammation. So how did we do that? Remember I told you that inflammation is caused by incoming monocytes and neutrophils, right? I told you that. What we did in this system was we actually added exogenous human monocytes to the system to recreate the inflammation. And then we were able to see cytokine storm. So here we would basically take the media from the 3D bioprinted and do an ELISA for IL-1 beta and other ones. So we could demonstrate inflammation, we could demonstrate cytokine storm. And then finally, for thrombosis, what we did was there is a very well-defined biomarker called thrombomodulin. And it has been shown that in COVID patients, the ones with the highest rate of mortality, this biomarker is actually seen at very high levels in the blood. So there's a direct correlation between COVID-related deaths and circulating levels of thrombomodulin. So we started to pick up on that and assayed to look for thrombomodulin as a biomarker for thrombosis. So we're pretty excited that we had the system up and running and going. And so the next step really was to then start for looking for drug repurposing, because that was the original intent. The original intent was not to create the Taj Mahal of a vascular lung, but the intention we had was much more focused. Can we identify drugs that are already approved by the FDA that could potentially be repurposed for COVID? That was the whole idea. So we started in collaboration with another company called Indras, who are experts in silico screening. So we took 8,000 FDA drugs, right? And through in silico screening, we narrowed it down to about 20 drugs. We went through different levels of filter before we came there. And then there were 20 that, based on about in vitro, the in silico findings, looked like they would be very good antagonists of the binding of RBD to ACE2. So the viral entry could be potentially done. And then what we did was we took these 20 and we screened them in our 3D human vascular lung model. The first thing we looked for was whether it's binding, blocking a potential drug is blocking the binding of RBD spike protein to spike protein RBD to AS2 or not. If that looked good, then check. We went to do the other things that are shown on the right-hand side in the small stick and ball model that I have here. We looked for anti-inflammatory effect of that drug. Secondly, we looked for cytokine, whether it will have any impact on cytokine storm. And then lastly, we look for the thrombosis biomarker. So this was kind of the funnel that we created using the 8,000 drugs. And we finally identified 3 drug families. And we're very excited to tell you what those were. Completely unexpected. See, this is the thing about doing an unbiased approach. Most people that were working have taken the available antiviral drugs and then screened them, right? So you will only find antiviral drugs. But our approach is more unbiased because here we could find drugs that potentially can do just many more things than just be antiviral, right? So we like this approach, and that's how we got into it. Just to tell you, the existing drugs, none of them were designed to fight COVID. As you know, they're simply being used off-label. Chloroquine, it was designed for malaria. Remdesivir, it was actually for, Ebola and all that. So all of these, and I fully understand why physicians did that, is because at that time they had no drugs and they needed, the sense of urgency was so much that they started to pick and choose what they thought would work and used it. But the important thing to remember is none of these were designed to actually combat COVID and were being used. And chloroquine, as you know, has been discontinued. That's what happens when you pre-assume something and start to push it. The data will drive you and let you know whether what you're doing is right or not. And chloroquine has to be pulled. Remdesivir is up and, the jury is still out, whether it helps or not. So long story short, just taking up existing antivirals or anti-whatever drugs are there, we didn't think was the right way to do it. We wanted to do it more unbiased, start off with 8,000, double-blinded, neither we knew it or the in silico company knew it, what to expect. And that was the beauty of all this, I think. So the first family of, and I'm not showing you data because it's all been published. If you go into bioRxiv, you will see and just put in SGLT2 inhibitors and COVID, you'll find it. So the first family of drugs we found that could be repurposed for COVID was very exciting. It was an anti-diabetic drug. I mean, it's completely unexpected. Again, if we had only stuck to looking at antivirals or anti-inflammatories, right, we would never have gotten this. So what are SGLT2 inhibitors? So these are basically anti-diabetic drugs that are currently on the market. They're available, they're not very expensive, they're available globally. So you don't have to rely on another country. You can actually, use this. So these, the way these work is they actually block the reabsorption of blood glucose by the kidney, right? And so the kidney secretes the blood glucose, and therefore the blood glucose levels go down. So it's a completely different mechanism by its work. We do not believe the SGLT2 inhibitors working for COVID has anything to do with the anti-diabetic effect. But as I'll show you, we found everything that we wanted in a molecule that we wanted to repurpose. It was found in the SGLT2 inhibitors. The other thing which is very exciting, it wasn't just that one SGLT2 inhibitor worked. The whole family, there's like 7 or 8 of them, as is shown on the table, that have been approved and out there. They're all available in India also, by the way. They all work. So there has to be some target they're hitting because it's a class effect, right? It's not just a one-off or a fluke. It's a class effect. So for a physician, he or she will have plenty of opportunities to pick and choose, which one to repurpose for COVID. Before that can be done, though, they have to go to clinical trials. So we have actually taken a pause here And we're looking to see partners. And I'll talk about what's out there in the clinical domain with this. But this was an outstanding, astounding finding for us, because we would never have expected. In diabetics who are already on SGLT2, there is anecdotal data that the frequency of COVID is actually lower than other diabetics, similar diabetics who are not on SGLT2. So that's anecdotal data. Our idea is not this to be, this is currently an oral drug. What we want to do is reformulate it as a nasal spray, because then, high concentrations of it can be sent to the lung. And also you will not have any associated glucose lowering effect in normal people. So where we are going with this story is we will develop a nasal formulation of an SGLT2 inhibitor and see if it works for COVID. And as I said, there is already some information from patients that this is a route that is likely to work. So very exciting finding for us. The other thing we found out of the 8,000 we looked at, the other thing we picked up was tyrosine kinase family of inhibitors. And the list is shown here. Most of these currently are used for cancers. And so, there's no reason why they should work in COVID, but that's what we found. Again, as I'll show you, they ticked every single box we would have wanted to see in a drug that could be repurposed for COVID-19. They are available, they are not very expensive, they're globally available. And again, if you want to look at this data, please go. Again, this data also has been published separately in bioRxiv, so you can go look at it. I'm not going to show you, but there's again, a physician has 7 or 8 different tyrosine kinase inhibitors that he or she could prescribe for COVID-19. Right. So this is, again, very unexpected finding we had. But as soon as we published this, we got this information that the US FDA has actually given an emergency use authorization for one of these. I think, but I don't know if it's not on the list, but one of the tyrosine kinase inhibitors has now been approved under emergency use authorization for COVID-19. So beautiful. For us, this was like, music to our ears, because that's what we would have predicted. And, independent of us, somebody else has done clinical studies and now gotten approval from the US FDA for this to be used. So again, a very good correlation between what we were finding and, the clinical studies. So one of the things when you develop a new model is it has to be it has to be predictable of what's happening in human disease, right? It's one thing to create it, but if it is not showing no connection with human disease, then it's useless. Then it's academic, then it's bioengineering. So this cannot be used for translation then. So here is just a simple, I just put together a very simple table to show you. And these are very early data. By no means does it mean that we have completed the validation, but the signs are encouraging. So SGLT2, does it block viral entry? Yes. Does it block, is it anti-inflammatory, meaning the monocyte adhesion and entry? Yes. Is it anti-thrombotic? Yes. Does it suppress cytokine storm? Yes. And what is the clinical correlate? I just told you. There is now data being published that in groups of diabetics, one set that is not an SGLT2 inhibitor and another set that you are on SGLT2, it appears that the ones that are on SGLT2 inhibitors, the propensity to get COVID, or even if they get COVID, they recover much faster. So there seems to be, we are starting to connect dots between our human vascular lung model and what happens in real life, right? Tyrosine kinase inhibitors, beautiful correlation. As I told you, just as soon as we should publish this, within a few days after that, now the US FDA has approved a tyrosine kinase for COVID-19, and it does everything we'd like it to do. So another very, very nice correlation, not proving that our model is highly predictive, but at least starting, we are starting to generate information and connecting the dots. There's another natural product that we picked up on. Homo harringtonen it has, and in China, somebody's actually doing clinical trials on this for COVID as well. So far, so good. What about chloroquine? We also looked at chloroquine. It actually shows some activity in blocking the viral entry into lungs. I think it's because it's preventing endocytosis, because it works by changing pH. But it has no anti-inflammatory activity. It has no anti-thrombotic in our hands, and no anti-cytokine. So if you had asked me a year back, would this work in COVID? I would have said no. And that's exactly what people are finding now. As you know, initially there was a lot of brouhaha about this, but now it's been withdrawn from the market. So it's a very small database. Of course, this has to be built out a lot more, but we are excited. We are excited because we are perhaps the only ones who have done this kind of work in the world. Understand the disease, what are the critical steps, can we create a model, and then screen drugs, and what happens in the clinic, right? The whole, the loop kind of thing. We seem to have been right on the money with a lot of things we are doing. So very exciting. So one of the questions I usually get asked is, why are 3D models more predictive of human disease versus, say, a co-culture or an organoid or a single culture? And, everybody has their reasons, but my reason is, first of all, the multiple cells are in proximity with each other, right? So like I said, in the lung tissue is heavily interspersed with blood vessels. So that's what we create. And as you saw, the layers that we created, right? There's a layer of lung epithelial cells, and right below that, as in real life, there is the blood vessel. So the architecture we are creating is similar to real life also. With other, like the organoid or some of these other technologies, you really don't have that degree of control on what you might end up with. And being able to bioprint in such a very precision fashion, because it's a computer robotized way of printing, we are able to get reproducibly also. The other things is molecules secreted by the two cell types can impact each other. So there's a cell-cell communication that can happen. The lung cell epithelial cells might secrete, or the endothelial cells might secrete TNF, and then it gets across to the lung cell, and it has its own, it might set off a different set of molecular pathways. So that ability to communicate with each other, I think, is a very important reason why 3D models are more predictive than others. And I already talked, as a drug discovery person, we have to have precision and reproducibility and a throughput. And I think 3D models, at least in the industry, are very popular for that reason. So is there anything else that needs to be done? I think there's a lot of work for us to do, right? First of all, if you look at our model, there's no blood flow. In real life, there's actually blood flow. So we are working on ideas on how to create blood flow. I think it's not that difficult. The other thing is we need to add other cells as well. There are other cells in the endothelium, blood vessel. There's also other cells in the lung. So we need to try and recreate some of that as well. That's going to go on. And then cell level sampling, can I just look at, even though they're all 3D bioprinted, can I dissect out the endothelium and look at the molecular signatures under different conditions? Can I do the same with lungs? So that's something else that needs to be done, to be able to just slice out that one layer. And this, I think, is pretty easy also for us to do. And then, we have to validate. I just looked at 4 readouts, right? There are many other readouts, like, for example, the black fungus, which I don't know anything about. But there are other readouts about the disease that we need to start validating as well. So a lot of work is ahead of us. But I think what we have gotten so far, I think, is really very, very nice. And as you can tell, it's starting to be predictive. And we are able to use this as a translation model. The end goal of our research really is to be able to convert anything we do into a translational tool that can allow us to select drugs better. I mean, that's where we are going with this, while as other people are doing other things with this model. So this is my last slide. take-home messages for all the students who are here. The 3D models of disease could really be the future of drug testing. And this is not just me. The industry is, the regulatory agency is also going in that direction. I think it's the European Union regulatory has said that no more animal models will be allowed after 2030 or 2040. They've given a date out. So everybody is moving in this direction. The use of animal models is not only unethical, as Brinda will tell you, but also it's not useful. I mean, we are unnecessarily using these models, and they're not as predictive. So the whole, I think the industry and regulatory agents are clearly married to the idea that we need to create more human-like models of disease. And 3D model seems to be a superior at this time versus some of the other models. Predictive value and validation will have to be done with a lot more rigor. I have just told you 4 examples, but clearly that's not enough. We would have to look at 100 compounds, for example, to really make a saying that, yeah, we are predictive. And then the way to do that, we are thinking is there is some retrospective clinical data available with certain drugs, right? So we already know what those drugs do in humans. So we want to take those drugs and see if we are able to get the same data or not. So that's the retrospective. In terms of the future, I think the skin, the lung, digestive tract are the low-hanging fruit. There are many people doing skin because, again, it's very nicely layered. The epidermis, the dermis, you can print it out and you get beautiful data. I think that is going to be the first one out the door in terms of being commercial, I think. The lung is another one that we are working on. For what we've done. We are also working on digestive tract, by the way. We are trying to create a leaky gut model. So there's a lot of diseases of the gut. IBD is an example that actually create a leaky gut and allow bacteria to get in. So we're trying to create a leaky gut model as well now. I think the more complex organs, such as the brain, which is multicellular, and you don't even know how to organize the architecture in a cell culture model, I think is very complicated. I think it's very, very complicated. Similarly, the kidney, to have a nephron up and running, working, that has many different things. People are attempting it, but it's not that easy. I also think, Dr. Radha, that liver is not as simple as we think, because again, there's so many cell types involved, I think, but these are the ones that are going to be critical in the future for transplant related research. So I think this is my last slide, and hopefully I'm good on time, so I'll stop here now and be happy to answer your questions. So thank you for your time. Yep.
And thanks, Dr. Uday, for the extremely wonderful and interesting talk. So we have several questions. We'll try to go through them, and let's see if time permits. So first question is by Shreyo Sengupta, and they're asking in inhalation toxicology, the focus has always been on systemic toxicology. We now have several lung alternate models, either whole organs, organoids, organ-on-chips, or models focusing on particular function of the lung. Do you think all of these models in combination can replace the use of animals? Are there any correlation studies done on these in vitro systems?
So I'm not an expert in inhalation formulation biology. To be honest, I don't even know what models are being used. But I think the critical thing for any model to substitute any of such models to substitute animal model is the correlation with what you observe in humans. You need to be at about 70 to 90% of what is observed in real life human beings and what you're getting. If you're able to hit that benchmark, then I think, yeah, there's a strong reason why the regulatory agency will say, yeah, sure, we can use this model and predict what's going to happen in terms of toxicity in the human. See, toxicity is a very tough, that one of the reasons we chose diseases is diseases are a little easier. Toxicity is a go, no-go decision tool with the regulatory agency. You might have the most beautifully efficacious drug, but if you're not able to demonstrate its toxicity, in a satisfactory, it's never going to move forward. So any tool that you are trying to create to get a readout on toxicity to convince the regulatory agencies will really have to be an order of magnitude better than what you can do with diseases, is my opinion. So the short answer, yes, but there's a lot of work that'll have to be done. Yeah.
Right. So next question is by Sandra. So she's the head of European Reference Lab for Alternatives to Animals. And her comments are great to see your in-house validation work and anatomy. And indeed, it is not simple. I would like to ask a visionary question on the potential of new applications of axial 3D, I think, imaging produced, which can produce a precision 1:1 scale 3D model, for example, of COVID-19 patient drugs from CT scans and how they can be used for determining the course of infection and routine applications in the clinic for human data generation.
Oh, I think that's a great area to be in, Sandra. I think, so if I understand what you're asking is you will take images from what happens in a patient and then use those images and recreate the lung, in silico lung, and maybe then a wet lung out of it. Yeah, I think that's the highest form of personalized medicine, I think. And I think that is a great area, and that's going to be realistic, I think, in the future. I have no doubt. In the dental field, that's already going on, as you know. Also for lungs, there's a pneumonia. People are able to differentiate between pneumonia and other lung diseases based on the images that they get. So yeah, clearly viable, and yeah, I think it's going to happen. It's going to happen soon. So yeah.
So next question is by Camellia Bhattacharya. They're asking, I just wanted to ask the possibility that human body might reject the model, as we have seen, like normal transplants are often rejected by the patient's body. So is it possible for 3D printed organs also?
Yeah, I think if you want to transplant an organ, you will have to harvest the cells from the patient, grow them out, then 3D print the organ and do it. So you cannot just take anybody's and do it. The only area in which you can do it where the rejection is very low is the cornea. And the cornea is also actually a layer 2. And there's one other company in India that's doing it. But if you want to do a kidney or a liver, yeah, certainly rejection is going to be a problem. And the way to get around that is to collect some patient cells, expand them externally, and then recreate the organ and put it back. Yes.
Right. So next question is by Ram Prasad. So they have 3 questions. So one is, can long-term effects of a drug can be tested using 3D models? Second is, in a normal immune response, usually there is fever, and fever can lead to temperature rise, which can then regulate fat metabolism and heat shock proteins. Can that be mimicked in 3D models? And third is, humanized models often involve reprogramming of somatic cells. So can we say that these 3D models are natural human models?
Yeah, so I forgot the first question, but
the long-term,
yeah. Sorry. Can you repeat that, please?
Sorry. The first one was long-term. Can long-term effects of a drug, can they be tested using 3D models?
Yeah, I think so. Although right now it depends on how long you can grow the cells, right? So certain cells, like the liver cells, are very tough. You can probably grow them for a month. I don't know, Dr. Radha knows. But I can tell you about the endothelial cells and the lung cells we've used. After about 2 weeks, it's very, very difficult to maintain. So you can never, ever do the long-term study unless the bioengineering reaches a stage where you can keep them alive for months together. I think it's going to be hard. It might be okay with certain cell types, but not all cell types. The other 2 questions, I'm sorry.
Right. So the second part was, in a normal immune response, there's usually fever, which can lead to temperature rise, which can then have an effect on fat metabolism or heat shock protein. So can this effect be mimicked in 3D models?
Yeah, absolutely. It can be. I mean, there are various ways to, the increase in temperature is really a conduit. It's a stimulus to excite the cells. So you can excite the cells, and you can induce heat shock proteins in 3D models. Highly doable. It may not just be, I don't know if you could just take the cells and warm them up to 30 9 degrees something, right? We are normally at 37. Can you warm them up to 40 degrees and see? That I don't know, but I'm pretty certain that if you have the right stimulus, you will see the molecular changes you're talking about in 3D bioprinted. Yes.
Right. And the third part was usually for humanized models, you have like reprogramming of somatic cells. So then are we sure that we have made a natural human model?
Yeah, see, I'm not a big fan of these reprogrammed cells. One, we have tried to do that with stem cells. Reproducibility is a problem. Batch to batch, we found huge problems. I really think that the Rolls-Royce or the Mercedes of this system is use patient-derived cells or primary cells and then 3D bioprint them. I think that is going to be the closest as you can get. Any other system like, using reprogrammable, I'm not so sure about that. So that's just my opinion. And that's based on some limited experience with stem cells that I've had. Yes.
Right. And then there are two questions which are related to surfactants, lung surfactants. So they say pulmonary pathologies often disturbs the surfactant production. And can bioprinted models mimic the physiologically relevant surfactant production? So,
yeah. Yeah, surfactants are basically phospholipids that maintain the tension that you need in the lungs. Um, I think it's reasonable to be able to do that experiment, and the only thing is, can they be maintained long enough to have good enough production of phospholipids? But it's worth a shot. I mean, it's an experiment that is likely to work based on my experience. Yes, I think it should be attempted. Yeah. We stayed focused on COVID endpoints. So that's why, we never looked at some of those things. Yes.
right. Then Dr. Radha is asking any ideas on mechanism of action. I'm guessing, Dr. Radha, are you talking about the diabetes angle? Why the anti-diabetic drugs are coming positive? Are you referring to that? Okay, I'm not sure she's there because
Okay.
she was like,
Yeah, I'll assume that that's what she's asking. And the honest answer is we don't know. There are two possibilities. One is it's completely unrelated to the glucose metabolism in the cell, which means we have a lot more work. The other is it is related to the glucose metabolism, right? So I don't know. Those are the two options available. We are going to look at the glucose metabolism is easy. You can change the amount of glucose in the media and recreate diabetes and see whether you get some of these effects or not. I mean, diabetes is one of the worst comorbidities for COVID, as you know. Diabetes, obesity, hypertension are some. So it's possible that it might be related to glucose metabolism, and that's why these anti-diabetics are doing something, or it's completely something very different, which is much longer. So right now, I don't have an answer, but I think it's a good question. And if she's willing to work with us, we can do that.
right. So there's another question on, has the cell signaling been characterized in 3D models?
Oh yeah, a lot actually. I haven't shown you anything, but if you go back and do some literature research, there's some beautiful comparisons between cell signaling pathways in cells that are grown by themselves versus cells that are 3D bioprinted versus organoids. So all that's been done. There's some very good reviews on that. And the short answer is yes, you can look at it, and it appears from that work is 3D bioprinted seems to have the closest molecular signaling pathways that you see in human organs. Yeah, a lot of data is out there. You can just go to Google Scholar and check it out. Yeah.
Right. And there's one question on, considering the very small size of alveoli vessel interface, how accurate is your lung model? What are the important biophysical characteristics that one would be missing due to this mismatch?
Yeah, that's a good question. And one of the things that we have to look at is what is the oxygen content? Are the oxygen levels different at the top cells versus the bottom? We haven't done those experiments. That's something we have to do. So we haven't done too much physico-chemical characterization, as you call it. That's something that needs to be done. But from a biochemical and molecular pathways perspective, we are pretty happy to see that what some of the drugs that seem to have worked in our model, people are now finding it's working in humans too. So at least from that perspective, the molecular pathways perspective, the tyrosine kinase story is very exciting, because within like a few days of us publishing this, it was, as I said, one of the tyrosine kinase inhibitors has been approved for COVID-19. So yeah, there is starting to be some connections, but we haven't done enough on the physicochemical. It's a good question.
Right. So actually, there are few more questions, but for the lack of time, we'll have to
Sure.
stop there. So one thing which we can do is we are while we are conducting the poll, maybe Dr. Uday, you could answer in the chat box if there are any, because there are also several more questions. So that is one option where you could type in the chat box answers to the questions. Second is we can send out your email ID and we can just type it in the chat box, and then anywhere they can write to you personally.
Yeah, I would prefer that they send me an email request. Surat, you're trying to make me do more work now. I just finished with 1 hour. So
Yeah,
please let them do some work and send it to me, and I'll be sure to answer it.
Yeah, sure. Yeah. So we'll just put in here because there are several questions. So yeah, because I think it was a very interesting talk. So I'm sorry, we'll have to stop there for this thing, but I'll just type it in the chat box, and Shyam, maybe we can.
Yeah. Thank you, Dr. Uday. Just before we move on, I think Dr. Suresh wants to tell something. So, Dr. Suresh, you can unmute.
Please.
Yeah, go ahead, Dr. Suresh.
Hey, Suresh. It better be good things you have to say. I'm just waiting here otherwise. We may have lost.
Yeah, Dr. Suresh, can you?
Yes, I just unmuted. Are you able to hear me?
Yes,
we
Dr.
can hear you,
Suresh.
Hi. Thank you for joining.
Yeah, thanks, Uday. Once again, I think it's an excellent talk. As always, you give excellent speeches. One thing I want to bring to the attention of the listeners is Uday and me also. I want to thank Uday because Uday and me have a combined project. I have a US grant for screening antibodies to COVID. And we have screened that antibody along with a drug conjugated to it. I think Uday is helping me with his model. I think that's an important point to notice because I think we are going to apply for a patent, and that could be as good as the anti-diabetic drug that was repurposed for COVID. So this model is helping in screening other drugs for COVID as well. That's what I wanted to say. And the next thing is I also worked on liver and lung and other organoid spheroid models. And actually, these two models can complement each other because there are some things that are lacking in this that are not lacking in the other. Obviously, the reproducibility is very high in 3D, but then the 3D interaction is very high in the spheroid. So I think I may have to talk to Uday about integrating these two. But again, congratulations, Uday, for an excellent talk. And thanks, Surat and the team for putting up a very good show again. Thank you.
Thank you.
Thanks, Dr. Pusala.
Thanks, Dr. Pusala, for the kind words. So moving ahead, as we had discussed earlier, that there would be a small 5-minute interactive poll session where we would like to know the general awareness on the good laboratory practices and good in vitro methods and practices protocols. So I'll just start the poll. So I would request everyone to kindly go through it and answer them. In the meanwhile, I'll also kind of put out the question and the options to the audience so they can also take their time and answer it. So I think the poll should be on right now. Can you guys see the poll?
Yes, I can see the channel.
So the first question is a straightforward, simple question. It's about the affiliation of you. So people can just answer whether you are a UG or a PG student, postdoc, scientist, medical professional, or a government body regulator. So you guys can answer it as in then. So the second question is, I see that people aren't participating. Surat, is the poll visible?
I'm able to see, but I'm not able to click. Brenda, are you able to click on the poll?
No.
Because I'm just able to view it. I'm not able to click on it.
Oh, just a second. Maybe I'll. Can you try now?
Not yet. Should I? Maybe you can end the poll and start again.
No, I think host and co-host, I think they might not be able to click, but I think I think people are answering in.
Right. Sure.
Okay. Yeah. Thank you. So I also request everyone to just participate and then fill in. The second question is on the awareness of the guidance document on good in vitro methods and practices, which usually forms the basis of in vitro testing and the methods and conditions to ensure that the results and data generated are rigorous and reproducible. So what do you think about the statement, and are you aware of these practices which are followed by using stem cells? The third question is on while performing experiments, do you have access to the requisite documentation regarding the origin of the cells and the tissues being used in experiments? So if you have any access to those documentations, so please rank them accordingly based on the availability of those. So the next question is on the do you think that the composition of media and or additives can have a role in reproducibility of an experiment? So if you think that it is likely or unlikely, so please fill in the response there. The fifth question is, or do you think that there are enough training workshops in India that address the relevant standards, regulations, guidelines, or the documents itself while working in vitro stem cell derived models? So usually, if there are students or postdocs, PhDs, so do we have any lessons or any sessions on these while coursework or any other courses where you have come across? So if you know any of those, so please mark accordingly. The sixth question is on the awareness on the legal and ethical issues which are associated while using human cells for biomedical research, such as informed consent of the donors and the rights which donors have when they are donating their cells for research or any other purposes, instances of approval or the process of approval by the local ethics committee, any other legal, national, international regulatory bodies or any other information on those. So if you have any information on those, so you can mark accordingly. And the last question is on the question of reproducibility itself. Do you feel that it is a significant concern in experiments related to stem cell derived models, MPS models, or other in vitro systems? So what do you think about it? Do you agree or disagree? So please mark accordingly. So maybe we will give it a couple of more seconds so that everyone can fill in. As of now, we see that almost 72% have participated. So if others can also fill in, then we can close it. In the meanwhile, Surat, is there anything else?
Yeah, so I've also posted Dr. Uday's email address in the chat box. So if anyone wants to get hold of him after the webinar for any further questions, so they can do that, or they can also write to us and then we can forward your questions to him. So either will be okay. And we'll also post the registration link for the newsletter again. And please do register for the newsletter if you're interested in both the research and also the regulatory practices and grants and any other aspects associated with human relevant research, both in India and globally. So please, yeah.
Dr. Uday, are you there?
Yes, I'm here.
Yeah, just because there's one question which kind of is there, that they're asking about the cost of a 96 well 3D inkjet based bioprinter. Are they currently available?
Yeah, no, we got this custom designed, and if you write to me, I'll tell you where. It costs a few lakhs rupees.
Okay.
It depends on, you know, what kind of bells and whistles you want, but yeah, write to me and I'll tell you where we got it made. Yeah.
Sure. I think we'll just give it a couple of more minutes. 95% have participated. So few are. So, and also I've also posted the link, the subscription link for the newsletter, for the CPHMS newsletter. So people interested can subscribe to it, and in a few days we'll be also coming out with the first edition of it, and we'll be also sharing them with everyone.
We can end.
I think we can end the poll. Once again, thank you all for taking your time and attending this webinar on a Saturday morning, and hope that we see you all in the next future webinars. Thank you all, and also we thank Dr. Uday for taking his time and giving us such a wonderful talk on in vitro models. Thank you, Dr. Uday, and
thank you, Dr.
thank you
Uday.
everyone for.
Okay, thank you very much, and thank you guys for taking time out. So, yep. And Surat, don't wait another year, okay? Okay.
Sure.
All right. Thanks a lot. I enjoyed it. Yeah.
Thank you, Dr. Uday.
Thank you, everyone.